I'm Hemdeep, and joining me is my co-host, Robin.
Hello. Yeah, so we are happy to be here on our second episode. Today we have Jack Kosh. Now, Jack Koch is an assistant professor at the Aquatic Germplasm and Genetic Resource Center in Louisiana State University Agriculture Center. His research focuses on developing cryopreservation pathways and germplasm repositories for aquatic species. Welcome, Jack.
Hello. Thanks for having me.
Can you describe your academic careers to date?
Yeah, so I did my undergraduate education at the University of North Carolina, Wilmington. I got a bachelor's of science in marine biology. And then I traveled across the United States to Oregon State University, where I got my PhD in integrative biology. And now I'm, like you said, an assistant professor at the Aquatic Germplasm and Genetic Resources Center. And when I arrived at the AGGRC, I started off as a postdoctoral research associate there for 3 years. And then I've been an assistant professor for just over 6 months now.
We had spoken a while back, and I think that was our first introduction. And since that time, I think I've done a bit of research with the help of Robin of who you are and all the work that you've been doing. I guess the question I've always had whenever I speak to researchers is to find out where that aha moment was, where they, you go through your undergraduate, postgrad, but where was that tipping point that happened for you where you really found your niche in the type of research that you're doing?
I'd say my path to where I am today has been a bit non-traditional in terms of the variety of projects that I've undertaken. For example, my PhD, I did some enzyme physiology, I did some genomics and some microbiology. And traditionally, when you get a PhD, and I'm not going to say most PhDs because everybody has their own journey, but you might focus on one thing. So your whole PhD might be in microbiology, your whole PhD might be in genomics. Mine was a little bit different, where I had a couple of different topics that my dissertation covered. And then moving to Louisiana, I had no background in cryobiology at all, which is a good thing and maybe, an opportunity for growth, because I didn't have this preconceived notion of what cryopreservation was or the processes that need to happen. I got to be trained by the group that I'm with now and got to figure things out on my own to form my own opinions and knowledge and information about that process. So the aha moment for me has always just been curiosity about the world around me. It started from a young age. We would go to the beach every year as a family. My grandparents, my aunts and uncles would come down, and most people go to the beach and like sunbathe or go swim in the water. But I had a seine net, and if you don't know what a seine net is, you should, maybe we can post a picture on the screen. But it's a giant net. They're about, I'd say, 10 feet long by 4 feet high. So they're pretty big, and there's posts on each side. In the bottom half of the seine net, there's some weights, and then the top half, there's some floats. And so I brought a seine net to the beach with me as like a 7-year-old, 8-year-old.
Sorry, did you yourself bring it, or was it like a family member had it and they introduced it to you, or was that just like you yourself?
I think I brought it. Like we would go into like the little stores near the beachfront, and I saw one, and I wanted to try it out. So my parents bought it for me, but I was the one who would bring it with us every year. And I would convince whoever would come out in the surf with me, my cousin, my grandpa, my grandma, my parents, whoever would drag the net to the surf with me, to come drag it. And we'd catch like fish and jellyfish and all sorts of things. So I think that's one of the main drivers why I'm so curious about the outside. But it's more than just an aha moment, I'd say.
So would you say that you lean on those experiences, or you leaned quite a bit on those experiences as you sort of decided where you were going to eventually sort of do your postgrad work or your PhD work, or even in further on as well?
Yeah, I guess I always knew that I wanted to end up near the coast.
Okay.
University of North Carolina, Wilmington. Their main campus is, with no traffic, it might be like a 10-15 minute drive to the beach, which is incredible. Like, you're right there. Um, Going to Oregon State University, we were a little bit further from the beach. It's like an hour and a half. And I didn't go to OSU because of its proximity to the beach. I went there for the person that mentored me in my PhD, Dr. Virginia Weiss. And she's incredible. I didn't know that at the time, but she's a world renowned biologist in the field of coral biology. And I'd read some of her papers while I was working on my honors thesis at UNC Wilmington, and I reached out, and things worked out well there. And then coming to Louisiana again, it's more about mentorship opportunity and training opportunity than it is like a location. And Virginia knew my boss, Terry Tiersch, Dr. Terry Tiersch, got to come see the facility and was like, Jack, you would really fit in well here. Like, you love building things and tinkering, and that's what they do. So combining, and we can talk about this more later, but combining biology and technology is a big thing that I have come to build my program through at the AGGRC.
Amazing. And I think Robin did a bit of stalking and found out that
Don't
you
call
were
it that, please.
Okay, sorry, bad choice of words. It's not stalking, but just did quite a bit of background dig on you and found that you are an avid wildlife photographer as well.
I
Or
am.
at least you used to be, yeah.
You used to be, okay.
I'm
So,
not sure if
what
you're
was that
still
about?
updated with it.
What was that all about? Like, how far did you progress with that? And do you ever do you use that as a moment of just being able to decompress from a day, or where is it now?
Yeah, absolutely. I don't get out and shoot as much as I used to. That could be because it's a little bit warmer in Louisiana and it's not as nice to be outside. But I wish I got out more. I still do get out. We had a once in a lifetime snowstorm here. We had 10 inches of snow in Louisiana, and I got out and took some really cool pictures of snowflakes. And I've nowhere near. There's people who take very, very incredible pictures of snowflakes out there, but I have pictures of Baton Rouge snowflakes, which I think is pretty cool. I'll share it with you guys so you can pop it up on the screen if you want. But yeah, I love taking pictures of wildlife, landscapes, the night sky, because it all fascinates me, and there's a lot of beauty out there. And yeah, it's totally a way for me to decompress.
Totally unnecessary, but a little shout out. I know for a fact that you also have a little store for your photography. So if anyone happens to be interested, there will be a link for it on our webpage, and they can have a look.
Yeah, I print all of my photos here at home, so it's not through a third-party service. Like, I have a relatively large photo printer here, and yeah, if anyone wants one, you're welcome to.
Wow. Look at this. This is so cool.
So you mentioned it a little bit briefly, but you joined AGGRC, and how long ago was that when you started with them?
Yeah, so it was right after COVID, sort of was right after people were coming back from remote work. So it was
I guess
February of 2021. Yeah, so it's just been 4 years now.
And what is kind of like your specialization around the lab? I know they said that you're a good tinkerer and you work with a lot of technology. So I'm going to assume it kind of leans in that direction, but you will give us a better idea.
Yeah, so they call me a corrupted biologist. My expertise is in invertebrate biology, and invertebrates are anything without a backbone. So you might think of jellyfish, sea urchins, shrimp, oysters, corals, things like that. And we have over 100 3D printers in our building, and we see the power of using 3D printing to progress our research and make it accessible to more people. And so my natural tendency to tinker with things very quickly merged with my biological background, and now I combine the two very, very closely. I called myself a technobiologist, and at the time that I said that word out loud, I don't know, 6-8 months ago, I didn't realize that anyone had defined it. But there's actually this really cool paper out of the University of Miami that defines technobiology. And in reading it, I'm like, wow, this is so cool that someone has defined this. And it's what I do. It's what I do. I combine technology and biology very closely. Sort of the opposite of that is if you switch the two words is biotechnology. And I don't remember what the distinction is at the moment, but like I'm not a biotechnologist. I am a technobiologist.
Interesting.
Wow. So there is a term for the skill set that you bring to the team. That's amazing. So now, if you were to compare your approach to solutions compared to other members of your team, how is it that it really differs? Like, where do you find your solution or your inspiration to finding a solution or challenges as opposed to other members of the team?
I'm in charge of the industrial scale portion of our facility, but we also see really important need to focus on the consumer side of 3D printers. Laboratories around the world asking someone to buy a $10,000, $20,000, $100,000 printer, that's tough, right? But if we can create hardware that advances biology on a $300-$400 consumer printer that you can buy off Amazon or even Walmart or any store that's nearby that might have technology like that, like that's a lot of power. And so my focus on the industrial side, we do rapid prototyping on that side. And so that's where I sort of think of things is how do I move through a bunch of iterations of a device as quickly as possible. And then there's other people in our group that are more focused on what we call open hardware, which is a piece of hardware that you can distribute as a digital file. And there's some freedoms that come with that open hardware, right? The freedom to use it, distribute it, modify it, share it. Sort of like open source software. You may be familiar with like the Linux ecosystem. That's a huge community that has come together to build this open operating system that you can customize it to your heart's desire and make it exactly what you want to fit your needs. And we see the same power in open hardware.
And so now the scale, you're right. Right now, 3D printing has a wide range of scale in terms of the $300, $400 range right up to $100,000. In terms of how you approach solutions, do you find that you need to run from the 100,000 and then try to scale it to a scale that it would be done at the $300-$400 range? Obviously, there's a significant gradient between what the deliverables are between the 3 to 100 range.
Yeah, absolutely. I like to say that the power or the limit of 3D printing is your mind, because you can do whatever you want with 3D printing. There's some technical limitations, but most people aren't going to run up against them. But absolutely, the capabilities of a printer are pretty closely tied with the price that you pay for it. Now, for us, we deal with a huge range of biological scale. So let's talk about that first. We think about whole groups of animals, which is like, giant things, all the way down to like sperm, which is a single cell, very, very tiny. And there's things that are even smaller than sperm, but we don't worry about that, right? There's like subcellular like enzymes and DNA things, right? So we're not focused on things that are that small. But for our needs, the scale of most sperm that we work with is within the range that consumer printers are able to achieve. Now, I don't know, hopefully your audience is familiar with what CADWorks does, but you guys make this really awesome printer that can do microfluidic devices. And traditionally, microfluidic devices are created by like soft photolithography that you need a multi-million-dollar clean facility, 6 months of time. And if you decide you want to make a change to your device, you have to go and do that all over again. And CADWorks isn't the only group that makes microfluidic. Right? There's a whole range of microfluidic printers out there as well. But the power of having a microfluidics printer is that you can prototype rapidly, and if you want to make a change to your microfluidics device, like we can pump out 20 different versions of a microfluidics device every single day to look at how different geometries, different sizes, different configurations of the microfluidic infrastructure change how, for sperm or cells, interact with what we're doing on the cryopreservation side. So that's just super incredible for us. But again, always trying to think how we can get back to consumer scale is super important for us. The last thing I'll make a note of here is the ability to 3D print molds for microfluidic devices. And that's a huge, that's moving towards the consumer side, the need of the consumer side that we are interested in. Because now we don't have to ask laboratories to buy $100,000 microfluidic printer, a $30,000 microfluidic printer, but we can print molds and ship them all over the world. And now people can make their microfluidic devices, but we can like rapidly create the molds for people to print on. So it's sort of just a matter of what your needs are in your laboratory, right? Do you need to be the one doing the prototyping of your microfluidic device? Or are you happy to, if you're a biologist, you probably don't need that. You just need the mold so you can look at the sperm or look at the cells under your microscope, which I think it's awesome that all of these new technological innovations have come online.
Yeah, you're right.
Speaking of like devices and the cryopreservation and everything, let's talk a bit more about your key kind of research topic, which is like the germplasm repositories, right? So like what is it? Like, how does it work? Just so whoever is listening to this episode, and not necessarily everyone is a biologist or marine biologist in general, so they have an idea of what it is exactly that you're working with.
Sure. So let's take it out of the marine biology context for a minute to something that's probably a lot more people are familiar with. But humans have been saving seeds for tens of thousands of years. We have corn. People are like, yeah, we can save corn. And the benefit of saving seeds is that you can plant them at an optimal time or place so that they will grow and make, rather than eating all the corn on the cob, if you save a few seeds, now you can make more corn in the future. The seed world or the plant world is much more advanced in time and technology for a variety of reasons in the germplasm repository world. So now let's talk about germplasm repositories in 2 words. So germplasm traditionally would be defined as any type of material that you can use to make a new organism, which strictly would be eggs and sperm. Now, we recognize that germplasm is actually a much broader category of types of cellular material that you can use to make new animals with. So eggs and sperm, embryos, larvae, cells, individual cells, because there is technology that would allow you to take nucleus out of one cell and put it into another cell. And you could make new lines with that, or new organisms. And then in some cases, you can even think about whole animals. There's a variety of different animals that are small enough that we can cryopreserve the whole thing. And there's also some animals that you can break pieces off of that animal and it'll just grow another animal out of it, sort of a clonal process. And then the second part of that is repository, which you can just think of as a bank, right? a bank is where you store your money. You can, they track it. They know how much and who has it. They know what type of money is there. And so a germplasm repository simply is just a bank of germplasm. It can be frozen. It can be living. It could be a museum sample as well, although germplasm is sort of not those would be like preserved samples, so you're not necessarily going to be able to create something new out of that, but it's just a place that you can store your samples, you can track them, where they are, you can put samples in, you can take samples out, and so that's basically what it is.
Is this a mandate that you and your team have, or is this something that it has been sort of mandated by a number of universities around the world, and each one is sort of adding to it based on the specialty of the program that they have?
So it's our mission to help people build germplasm repositories, help them develop cryopreservation technologies for aquatic species. And there is no mandate for us to do this, but we see the need for it. And you would think that the value, the economic, let's just talk about economic value for a second. The economic value that, let's say, oysters or salmon or trout or your favorite seafood, right? The economic value that brings in is enormous. And you would think that we would want to protect those genetic resources, right? Mm-hmm. We spend a lot of time picking the right oyster that we want to grow because it has disease resistance or it grows the fastest. Same thing with fish, right? People pick a brood stock of fish that they want to take those genetics and move them forward in life. You would think that we have those all protected, but we don't. We're very, very far behind in the aquatic field, especially compared to plants. The dairy bull industry is a multi-billion-dollar global industry of preserving the genetics of bulls and distributing all over the world. For aquatics, it's just not there. So there's no mandate, but we see it as a very important mission, and everything we do is in alignment with that mission. Now, there is a push from outside groups to make it a mandate. Right? When you submit a grant to a funding agency, it'd be really nice if that funding agency said, hey, you're going to be creating novel genetic resources for whatever reason. We want our investment, like we're going to give you $1 million, right? We want our investment to be protected. So you need to have some sort of plan of how you're going to protect it. Because so many times, there's research done on a line or some sort of genetic resources created, and there's no way for somebody in the future to go back and access it. It's not that it's like completely lost, but no one maintains it or protects it in the way that we would a plant or the bulls, right?
And do you know if this is this preservation is being done on a wide range of terrestrial animals, or is it now outside of the bull or maybe some other livestock? Is that really where it's relegated to right now? And you're sort of taking the mantle for aquatic, and then there's possibly a team that is probably going to be doing it for terrestrial animals, so on and so forth.
I'm not I don't have as much information to give there, but my guess is that terrestrial is a lot easier because we're terrestrial animals, and our knowledge of the terrestrial animals' reproductive biology is probably a lot better compared to aquatics, because we can't just go watch a fish all the time to learn when it spawns, right? Whereas we can watch like other terrestrial animals a lot easier.
Is that like lack of observation kind of one of the reasons why the aquatic wildlife, like repositories for aquatic wildlife, is so far behind?
I mean, that's probably a contributing factor, is lack of observation. Probably also like a lack of like knowing when to observe.
Just no one bothered to do it.
Yeah.
So when did this your kind of research start popping up in that case?
I'm not sure what you mean.
So like this whole research topic of building up repositories, trying to find ways to just preserve the genetic material of aquatic species. When did that start happening? Is it like in the past decade or even less than?
Yeah. Our center director, Dr. Terrence Tiersch, he started thinking about this a couple of decades ago, and he's been pursuing this for a very long time. He's worked with hundreds of aquatic species in the aquaculture industry, wild fisheries, conservation, and more recently, we've been working with some biomedical species. But he's been working with all over the globe. So he's been working for a long time with it, and it's really he's really been a world leader for a very long time in it. And there's other people who've worked in it. It's just putting it all together and seeing it at a global scale and global need. Terry has been very instrumental in that.
And if you guys were to sort of have a set of milestones for this repository, where is it in terms of milestones? Obviously, the number of species that you could sort of foresee yourself itemizing and then having a repository for it, it could be very large. But do you guys internally have sort of a number or the type of species, or do you classify species that are important for this type of safeguarding, and then those that anything like that?
We don't have any internal classification of species, nor do we have like a number, because there's a lot of aquatic species out there. We work pretty closely with the United States Department of Agriculture National Animal Germplasm Program in Fort Collins, Colorado. And that's a world state-of-the-art germplasm repository for animals. And I think they also have a plant section there as well. The plants are distributed. The plant germplasm repositories are distributed in a network across the United States, whereas animals are centralized there in Fort Collins. So they probably have a more curated, might be the wrong word, but more curated goals of what they would like to see in the collection there. I know that more recently people have been concerned about the two camps of people, like just collecting samples just to have them, so a high number of them, versus the quality of the samples. So collecting samples that are of strategic importance to whatever field that they may be relevant to. For example, if we take oysters as an example again, there's many types of lines of oysters because people grow them in different areas. You might, different farms, different hatcheries. We could ask each of those hatcheries to send us a couple of oysters, and we freeze their germplasm and store it there. And now we have hundreds of lines of oysters. Or we could say, hey, send us the 5 oysters in the United States that are super disease resistant. And so now we only have 5 lines, but the quality of those lines might be perceived as being a lot better than the 100 lines that could be out there. And I don't think there's any right answer between quantity and quality. Yeah. It's all about what the mission of that particular project is, because in biology, a lot of times quantity is a good thing, especially when you're talking about genetics, because you can get things like genetic bottlenecking, where you take 100 genes and you're doing a lot of breeding and you end up with 2 genes. And now your chances of like inbreeding go way up. So I don't think there's any one right answer between quantity and quality, but it is something that you have to think about.
Going back to like barriers, right? For why it didn't advance as far as it has as of yet. Is there like drastic changes in like the methodology to cryopreserve certain materials, or like between animal species? Like, I guess it's different for the sperm versus the egg versus the embryo, that sort of thing.
Yeah, so we like to focus on different levels. So there's like a protocol level, which would be like add 5 milliliters of cryoprotectant A to your sperm sample. So very specific. But then there's a level above that called the process level. And at the process level, most cryopreservation is the same, whether you're
Okay.
doing it for sperm, eggs, embryos, larvae. There's some differences, but at the process level, a lot of things are the same. And it's just a matter of fitting that process to whatever environment you're working in or what material you have available to you.
Okay.
Now, those specific types of germplasm, sperm is the most advanced because it's a single cell, it's small, and it's abundant, right? Usually, when you collect sperm from something, you have millions or billions of sperm. And if you lose half of them during the cryopreservation process, like it's not a big deal because you still have millions more. Eggs are really far behind because they're typically pretty rich in lipids, they're pretty fatty. And so freezing them is just very difficult.
Right.
And then larvae and embryos are rapidly advancing we're rapidly advancing in our technology and ability to cryopreserve those. And even bigger things like whole organs, people are figuring out how to freeze like livers and kidneys, things like that, that will advance the medical field a lot. But it's a slow process to figure things out like that.
Okay. Because you have two papers. Well, not, you have way more than that, but I'm going to refer to two. One, preserving the, oh my God, I don't know how to say this, but Hydrocytinia symbiolongicarpus. Who needs tongue twisters when you have scientific names?
Hydractinia.
Hydrocytinia. Okay, so
Yeah.
that sperm and the California sea hare eggs. So if beyond a certain level, the cryopreservation is similar, like what aspects did it differ between these two specific species that you worked with?
So Hydra are pretty cool. They are like miniature. Have you guys seen Finding Nemo?
Yeah, of course.
You know the sea anemone that Nemo lives in.
Mm-hmm.
Imagine that sea anemone, but like a couple millimeters tall, and imagine that there's like hundreds of them on a single microscope slide. A microscope slide's like this big, right? That's how big these Hydractinia are. And so collecting sperm from an individual is very difficult because they only produce like a couple microliters. And so for that process, that's the sperm process. And sperm is relatively easy. We had to address the barrier of how do we collect enough material to actually freeze. And so we created a little open hardware device that you can put multiple slides together, and when they spawn, you have a nice soup of sperm rather than you get a couple milliliters of sperm instead of a 1 microliter. So Hydractinia is the sperm side. Aplysia are interesting because they're simultaneous non-selfing hermaphrodites, and they do internal fertilization. So let's unpack that. So simultaneous hermaphrodites mean that they have both male and female reproductive biology at the same time. Non-selfing, they can't take their own sperm and fertilize their own eggs. And then internal fertilization, so they're not like broadcast spawners. They actually, they copulate with each other. They form these chains of these mating chains. You can get like 20 individuals where the first one is the female for the second one, which is the male, who is the female for the third one, who's the male, and so on and so forth.
Wow.
And so for Aplysia, collecting their sperm and then figuring out how to do internal fertilization was one option. The other option was working with their egg masses. And it looks like a ramen noodle or a big wad of cooked spaghetti. It doesn't taste like spaghetti, but it has the consistency of cooked spaghetti. And we'll definitely need to put up a graphic to hopefully describe the complexity of this, because if you zoom in on a single spaghetti strand, it's not just a bunch of embryos living in there free floating. There's capsules within the egg strand, hundreds of capsules, thousands of capsules within one of these egg masses. And each capsule can hold up to 100 embryos inside of it. So one egg mass, like the size of a baseball or like a fist, has millions of babies in it. Millions. Which is all part of their reproductive biology, where you make lots of babies and only like 1% of them make it to adulthood, right? That's a normal.
So this egg mass, is that from the entire chain of the sea hares, or is that like from one pair, for example?
That's from one individual makes an egg mass. Yeah.
Oh, okay. Wow. That's a lot of
I
eggs.
I can see how the whole notion of having a camera in the middle of a pond waiting for these things to spawn would not serve you well.
Yeah,
you would never know what on earth happened.
And we're lucky because Aplysia are intertidal, so they live in the zone on the coast where low tide goes out and then high tide comes in. So during low tide you can go out there and like watch them, but for like animals that live in the middle of the ocean, what are you going to do? Hope that your boat is in the right place when they're spawning? I mean, there's plenty of stories of animals like great white sharks. We for the longest time we didn't know like where they gave birth because the ocean is massive and good luck. You just have to be there at the right time or have the right technology to track them. So we're lucky in that they're in the intertidal and people have a pretty good understanding of what's going on in the intertidal zone.
Wow.
So these open hardware devices that you're developing, I guess that's for multiple, it goes across the entire process. So from collecting the genetic materials to, I guess, loading it or preparing it for cryopreservation, right?
Mm-hmm. Yeah. So it's really cool. If you look at the evolution of our open hardware technology through time, at first we were creating a single device that would address one step in the cryopreservation process. And now we're introducing a kit concept. Have either of you gotten like HelloFresh or Blue Chef? They deliver
I'm aware of what it is.
Food kits to your door. Imagine that for cryopreservation, where you get a kit to your door. It has everything you need to cryopreserve your sample, and that's what we're working on right now. Is creating these. We're calling them capability kits right now that allow you. They provide you the capability to do cryopreservation. Our first one will be for axolotl salamanders, I think. But one of the great things about open hardware is that anything we create is pretty generalizable to any species. So we're not just creating a piece of hardware for axolotls and a piece of hardware for frogs and a piece of hardware for Aplysia sea hares. We're creating one piece of hardware that can be used for all of them.
Does the hardware need to be tweaked in any way to cater to specific species, or exactly the same, like standardized across all sorts of species?
Standardized across, let's say, most species.
Okay.
So what is the key characteristics that you use to standardize across all these species? Like, what is the key characteristic that each of these species have that allow you to sort of use that one template right across that entire range?
There's a couple of ways that you can generalize or standardize by. You can do it by germplasm form. So whether you're working with sperm, eggs, embryos, egg masses, tissue, right? You could split by that. There are some devices that in theory could work with any type of germplasm. The other thing that you could split by is the type of cryopreservation that you do. And there's two major types of cryopreservation that people use. And there's a slow cryopreservation that the freezing rates are anywhere from like 1 degree C per minute to 60. And then there's ultra rapid cryopreservation called vitrification. And the freezing rates or the cooling rates for vitrification are thousands to tens of thousands degrees per minute. So it's very, very fast. And so there's probably some you could split these devices into two categories based on that as well. There's probably other ways that you could standardize devices by different categories as well.
Wow. And I think when you talk about the rapid freezing and those two techniques, I think the thought that crossed my mind was, there are humans that want to be cryopreserved, and what does that look like? Like, what is which one would they do? Wasn't Walt Disney cryopreserved, or wasn't there someone very famous as cryopreserved?
Possibly.
Yeah.
There's a
So
whole
which one
field.
would that have been?
I'm not sure. I'm not versed in that field. So, but there, it's called cryonics, I think.
Yeah.
And so there's whole groups of people that are interested in that. But yeah, I don't have much that I can say on that.
Sorry about that. I do get these moments of just epiphany, and I need to sort of sometimes just vocalize it out.
That's fine.
Okay.
So can you actually explain more about what exactly is in your cryo kits, and like, have you already started distributing it to, other people in the research community?
Yeah. So if you just imagine, it's a cardboard box and it has different modules in it. And each module addresses a certain set of steps in the cryopreservation process. So there's a module for collecting time and temperature data, which is very important for cryopreservation. That time and temperature data is quality management, so that we can make sure that that their samples didn't get too hot, or that they cooled at the rate that they said that they cooled them at. So many people do cryopreservation, they don't record the cooling rate. And you're like, you just have to trust them that they said, my samples were 5 centimeters above the liquid nitrogen, but the box size matters, and whether there's anything between the liquid nitrogen and the samples, that all matters. And so providing someone with a tool that they can actually send us a piece of data to be paired with the sample now makes that sample that much more valuable. So there's a module for time and temperature. There's a module for filling and sealing and labeling French straws. And French straws sort of look like coffee stirrers. They were created by the dairy bull industry. A lot of what we do relies on all the technology that's been created by the dairy bull industry. We're not reinventing the wheel. And when we want to scale up, right, if we want to freeze 100 straws or 1,000,000 straws, there's a lot of technology that we can use from that field. There's also a special box that is customized for each species or each group of animals that the kit is for. So one of the animals that we work with right now with the National Institutes of Health is the axolotl, or the Ambystoma salamanders. You may be familiar with them from How to Train Your Dragon, the key dragon, I don't remember, Toothless. Toothless is based on an axolotl. And now if you go look up axolotl and then go look up a picture of Toothless, you're like, wow, they are remarkably similar.
He has little ears, I guess. Yeah. I don't know what they are, ears. I'm just calling them that.
And then Minecraft is also like exploded popularity of axolotls. But anyways, I digress. There's a special box in there that would help you collect germplasm from axolotls in a standardized way. There might be a special device that we create for oysters or for salmon that we could package in there. It's a customizable module. And then there's the meat of cryopreservation, which is the freezing or the cooling process. And there's a module that provides you a device to help you cool your samples, again, in a standardized way. In any laboratory, in any field setting, there's a module that will let you do that. And then we were really fortunate to find that there is a pretty accessible shipping device. It's called a dry shipper. And the way a dry shipper works is you pour liquid nitrogen into it, and it has absorbent material in it. So it absorbs the liquid nitrogen into that absorbent material. And rather than there being liquid in the bottom of it, it's just stored in there, but it keeps it cold. And you can ship this anywhere in the world. If you don't have access to liquid nitrogen, you can charge it up, ship it somewhere, and do your cryopreservation. but they usually cost, I'd say more than $1,500. Again, that can be pricey for some parts of the world, and having something that is cheaper, more accessible to more people is powerful. And so we found a dry shipper that is a couple $100, and we've adapted some of our open hardware technology to be usable with this dry shipper. And so altogether, this kit delivered to your door is supposed to, the vision for it is that it's going to let you do cryopreservation wherever you are in the world. If you're in a laboratory or in the middle of the jungle somewhere.
That is amazing. So now, in terms of the timeline, when you and your team are hoping to get it out there for a lot of the other researchers, what does that look like?
So we're in the beta testing phase right now. So we've sent some kits out to people that we've collaborated with in the past, and we're asking them to break it. We want to know like what doesn't work, what does work. The biggest thing that we've been working on right now is the instruction manual. And I mean, cryopreservation is not like the easiest thing in the world. It seems like the best way to help people understand cryopreservation is to make sure they feel confident, is to make sure that they understand why they're doing the processes, understand the physics and the chemistry of cryopreservation. And that's really powerful because you are empowering them so that if they run into any problems, they can troubleshoot and make decisions that are smart. And so how do we have an instruction manual that conveys the necessary information in detail that people want, or detail that people don't want? And so that's been one of our biggest pushes right now, is trying to get some instructions out to these beta testing groups. So timeline, I'm not sure, but if you're interested, let us know. I think we might be working on like an interest form where we could find out what species people are working with, where you are in the world, what your experience level is, so that we can gauge what the need is. And I think in the future, because we're not a huge group, right? There's 8 professional staff members there, and then at any one time we have like 40 undergraduate student workers that help us on a variety of different research, husbandry, maintaining the building tasks. And so like we're just not capable of printing hundreds of these kits. And so eventually I think the vision is that it'll be commercialized in some way. I don't know if commercialized is the right word there, but we won't be the one sending the kit to people. We'll focus on developing the biological background, new devices to go into the kits, and have somebody else distribute them to people.
How does this kind of fit in with your open hardware? Because the open hardware is a digital file that they're able to 3D print themselves. So, and the Cryo kit is a physical product. Is there kind of like plans to be able to turn it into a kit that you can just fully 3D print, or whoever is getting this, they're able to 3D print everything by themselves?
Mm-hmm. Yeah. So that is probably one way that we would provide the kit to people, is we can, and we do this. If we go back to the example of the microfluidic devices, we can print the microfluidic devices themselves for people, which would be, potentially very difficult. We could send the molds to people and let them mold as many devices as they want. We could send the files to the people, and they could just print and modify them there. The same thing can be applied to this capability kit, and that we can send you a kit and that has everything ready to go. You don't have to do anything. We can send you the files for the kit, and you can go print it, right? So absolutely, like there's open hardware is very tightly interwound with this kit, and there will likely be many different ways that people can access the kits.
Makes sense. I had a question, but I forgot what it was.
I can jump on. So there seems to be, I guess these kits and this open hardware has a series of different type of modules or devices. Can you describe some of them? There's one that I've read up. It's called the Strand Cryopreservation Shuttle System, or the single sperm counting chamber. Like, first of all, how did you develop those devices in terms of the brainstorming or the ideas that came up that were used to develop them, and then how did you get it to a commercial object at that point?
Sure. So let's start with the single piece sperm counting chamber, and that evolved from a need to give people access to a way of counting sperm, finding out the concentration. So just like we talked about, time and temperature is an important piece of quality data that goes with your sample. Knowing the concentration of the sperm, if it's motile, what the motility information is, those are also pieces of important quality information. There's some commercial options out there. There's something called a hemocytometer, which was developed for counting blood cells. There's also something called a macular chamber, which was developed for counting sperm cells. Those cost like $300 and $700 each. And so if we can make something that's 3D printed and costs, 10 cents, 20 cents, $1, that someone can print anywhere in the world, that will allow them to count sperm repeatably and standardize across many laboratories, like that's wonderful. And so that's where the single piece sperm counting chamber came from, was this need to have some sort of device that was low cost, accessible to people anywhere, that could be packaged in a kit like this. The Strand Cryopreservation Shuttle system also came from a need that we recognized in trying to develop cryopreservation processes for aplesia. Before I talk about the shuttle system, though, when we work with outside groups for this project, for the aplesia project, we work really closely with the National Aplesia Resource, which is based out of the University of Miami. And so many times people, including ourselves, have created a device that we think is going to be useful for a community. And then the community, you give it to them and they're like, we don't need this. Like, that's not a problem. Or that doesn't fit into our workflow. Like, that's going to cost us another person. And so we work really closely with these national centers in something called user-centered design, where we form relationships with them. We talk to them. We ask them questions. We go see their facilities. We make observations. And we find out what types of hardware are needed to help them in their facilities, especially as they move to incorporate cryopreservation. So the Strand Cryopreservation Shuttle system is a perfect example of that. So we have many meetings with the folks at the National Aplesia Resource. We've visited their facilities. We make observations, and then we bring it back to the AGRC. And while we're developing cryopreservation processes for aplesia, we keep in mind what their needs are, what the environment is, and that's what this came out of. So to give you guys a mental idea of how difficult it is to work with aplesia egg masses, imagine you have that coffee straw and you have a little piece of wet noodle, and you need to package that piece of wet noodle into the coffee straw repeatably over and over again.
How many eggs do you need in the straw, or noodles do you need in the straw?
Let's say you want to put one noodle per straw,
Okay.
1 centimeter piece of noodle per straw, and you have 100 straws, and you have 10 minutes to do it. Like, it's not possible, right? And then if, one centimeter piece of noodle in a 20 centimeter straw, that seems like a huge waste of space, right? So wouldn't it be better if you could package multiple noodles in the straw? But now if I asked you to package 3 noodles in 100 straws in 10 minutes, you're like, Jack, 100 wasn't possible in the first place. Like, you want me to do 300? Like, no. So we created this device that, and I'll send you guys a picture so you guys can put it up and so that the audience can understand. But you take this device, you put your egg strand pieces into a set cassette, and you take this device and you just push down and you load 10 egg strands at a time into these cassettes, into these shuttles. And now I can do something with them. I can take these 10 and I can put them back in the seawater. I can load, more. So I could load 100 egg strands ready to go. And now I'm not on the clock, right? I haven't started my 10-minute timer. I can take those 100 strands that are packaged, ready to go. I can start the clock for them. All right, I have 10 minutes. But now I take my straws and I put them in something that has them in the same spacing as these shuttles. And now when I'm ready, when my 10-minute timer is up, I take and I just snap 10 off at a time. I can do three, and now I can get 3 noodles in each straw, and my packaging efficiency goes through the roof. So that's the basis of this sort of system, is just efficiently sample handling and packaging things. And then it also, we developed a way to seal those straws. Traditionally, you would use an ultrasonic sealer, which uses like high frequency vibrations to like melt the straw closed, but without heat. An ultrasonic straw sealer costs $10,000. And so in most parts of the world, including the United States, if people don't have access to that, they might take like a pair of tweezers and heat them up over a flame and use heat to seal their straws. But heat's not good for sperm, so you probably are affecting the sperm in some way sometimes.
Right.
So we made something that we can 3D print. We have 3D printed sealing balls. They interact with this system, so you have 10 straws that are spaced appropriately. You have 10 sealing balls, so now you can seal 10 at a time. And again, rapidly increasing the time, or decreasing the time that it takes to fill, seal, and prepare them for cryopreservation. And you're increasing access, because these are all printed on a $300 consumer scale printer with basic resin. Like, it opens the door to so many possibilities, not only for aplesia, but for other animals all around the world.
So for a lot of these devices, did you immediately jump into using 3D printing as a means to fabricate these, or were there experimentations with other fabrication methods?
While I've been there, we've only used 3D
printing. Oh, okay.
Yeah.
And what was the previous, before 3D printing, those cassettes that you mentioned, or the straw that you mentioned, was there a tool set that was used for this, or is it something like, is this all just a brainchild of necessity, I guess?
Yeah, it came out of my brain one night. Yeah. And that just goes back to the limits of 3D printing are your imagination, because you can print pretty much whatever you want. It's incredible. Yeah.
But even then, like, I guess the 3D printing is not like at its peak yet in terms of technology. Were there any, or do you find that there's still kind of limitations that 3D printing has with like your research?
Yeah.
And what would be the main ones?
Printing for cryopreservation, reducing the amount of thermal mass around your sample is very important. So if a 3D printer can only print something that is, let's say, 1 millimeter wide, let's say it can't print anything smaller than 1 millimeter, I have to have a 1 millimeter thing next to my sample. Let's just take this, and that 1 millimeter thing holds heat at some amount, right? And so if there was some way for me to cut it in half, so half a millimeter, and still have the same structural integrity, like that would be great. Mm-hmm. And that's what we see a lot, is just being able to reduce how much is there while still having the same structural integrity. And maybe this is when we bring in engineers, right? I'm not an engineer, but this is when we bring in engineers and they say, yeah, if you add some cross links in the back, you get that rigidity back and you can reduce um, how much material you have. There's a huge, like the technology is growing very rapidly for like metal 3D printing and polymer 3D printing, especially on these smaller scales. And metal is great because it's a heat sink. So you can rapidly pull heat away from your sample much better than you can with a traditional resin. Incorporating things like, oh, now I'm blanking on the name of the material. Oh, graphene, like different types of mixing resin with these different materials to increase its ability to cool samples quickly or its structural integrity. And then toxicity of resin is something that people think about a lot. Or I guess toxicity of any sort of material, even the plastics that we use in the fused filament fabrication printing. You just have to do a test, right? And a lot of the times your sample may not be in contact with that device for more than a couple of seconds, or you're using a sub sample of your sample and you don't care if it ends up non-viable in the end, right? You have your sample that you're freezing over here, and maybe you're running a sample through a microfluidic device, and it's just going to run through. You're going to do some sort of analysis in 2 seconds, and then you don't care about that piece of sample anymore. So, I mean, those are some of the things that people think about.
Yeah. And so you said that the AGRC has what, over 300 3D printers, right? Something like that. And like, how does that look? Oh, sorry, carry on.
No, I think we might have just broken 100.
Oh, sorry. Wow. I'm totally misheard that. Well, I guess okay. 100. But anyway, that's a lot of printers. And how does that look like? Is this all in like one gigantic room or warehouse, or how does that work?
Well, you guys will have to come down to Baton Rouge at some point to come visit our center. But we have a printer farm, which is one big room with maybe 50 3D printers in it. And they're just on shelves, right? If you go look up on a printer farm online, that's what we have, is a bunch of 3D printers on shelves. But we have printers in many other rooms. So I have a couple of 3D printers in my office. We have a training and prototyping space, which has about a dozen 3D printers in it. Anybody who comes to the center to work, any student worker, no matter what their major is, they learn to 3D print. And so we have this training and prototyping room where people learn to break printers. They learn how to fix them. They learn how to do their first print. They learn CAD. They learn slicing. And then we also have our industrial space, which is at the other end of the building. And that has a room that has 6 or 8 3D printers in them. And then another room that has a couple of resin printers and a polymer printer in it. So they're spread out in different places. And It's usually based on like what type of, what level of 3D printer it is, and then what its purpose is.
Mm-hmm. So because talk about the AGRC a little bit more. Clearly, 3D printing is very important for a lot of the research that's going on there. But maybe you could talk a bit more about like what the AGRC kind of, its mission essentially.
Yeah, so I mentioned it earlier, but we help other people and communities around the world form germplasm repositories. And that includes thinking about the process of cryopreservation, technology development, how do you bring other users in, where you're going to store the samples, how you're going to track the samples, how many people you need, what type of equipment you need. So that's what we help people do for aquatic species specifically.
And I remember in our first introductory meeting, you mentioned something about 4 pillars of the center. Like, what is that, for example?
Yeah, so we support our mission through 4 major programs. The first one is basic biological research. So we we like study animals to learn about their biology, reproductive biology, or evaluate how different chemicals interact with their germplasm to prepare them for cryopreservation. We have a technology development program, and that's sort of split into open hardware and industrial processing. We have outreach because it's super important to talk to people in other industries, other universities, other levels of education, to not only improve how we think, but also let people know that these technologies and resources are out there. And then the last one is industrial scale processing. We could run a company out of our building. We have the equipment necessary to double or triple the total global aquatic species germplasm samples just in a couple of weeks. We could double or triple what's out there. Obviously, we'd need enough sperm to do the freezing of, but we have some high level equipment that we do use for certain projects, and we also help we train people on that sort of equipment as well, because at some point people want to go from 10 to 100 samples, 100 to 1,000,000 samples, and there's different levels of scale that need to be recognized and thought about. Because 10 samples, you might be able to hire one person. 100 samples, maybe you need two people. But when you get to 1,000,000 samples, like, you can't just hire more people. You need some technology to help you accomplish some of those tasks.
In terms of, I guess the center has grown significantly, it sounds like, and it's quite important for this sort of repository. I'm wondering, how did the growth of the center sort of follow the trend line of the adaptation of 3D printing? Did it grow hand in hand? Was it a necessity? Or if you were to remove 3D printing from the equation, was the center had its own inertia that it was going to meet these mandates that it had set out for?
The center definitely would have continued without 3D printing, without the 3D printing intervention, but the growth has been much more rapid and has allowed us to address more communities, more diverse communities in different places than if we didn't have 3D printing. Because there is a big commercial, there are many commercial technologies out there for cryopreservation that we probably could have used, but giving it or making it accessible to people all over the world just wouldn't have happened as readily. When 3D printers became accessible to more people, I don't. I'm going to put a number out here, but I don't remember when the first 3D printer was purchased at the center. Maybe, let's say it cost $1,000, and that was like a big turning point, because they used to be a lot more than that. They 3D printed something, ran down the hall and dunked it in liquid nitrogen, and to see if it would still stretch, and it stretched, right? Usually you put something in liquid nitrogen and you try to like bend it and it just shatters.
Mm-hmm.
And that was a huge realization to the people at the center at that time, was that, wow, we can make devices that are going to be near and in contact with liquid nitrogen, and we can bring it to more people and make standardization and access just we can make it happen. So absolutely, 3D printing has driven or helped direct where we go along our mission.
And the community that you're trying to bring this to, do you find that they already have experience with 3D printing, or is it something that you kind of teach them about?
It's a mix. There are some people who know about 3D printing but have never done it. There are people who have like printed one or two things. There's people who have never heard of it or seen a 3D printer before. But more and more local libraries have 3D printers. A lot of universities have 3D printing facilities that their students are learning on. Any university with an engineering program, I'd say, typically has some sort of 3D printers. there's a lot of groups of students that are forming like maker clubs that have 3D printers. So more and more people know about it. But we don't necessarily like teaching people about 3D printing. We like helping them and making the environment such that they can succeed in 3D printing, because we find a lot of power in having people learn on their own while providing support for them. Like, if you have questions, like, we're here for you, but we're not going to tell you what buttons to push. I mean, we can help guide you, but we're not going to like give you some recipe or some formula, because that's not going to work every single time, right? 3D printing, you need different parameters for different things. And so having an innate understanding or a learned understanding of what those parameters mean, or really helps people progress as they move along. And we can't help every single person either.
Yeah.
Right. we need it to be a network approach where we help one person, they help two people, and eventually everyone has the same background.
And I guess that's why it's so important for your team to like bring this more to the commercial scale as opposed to industrial 3D printers. And I think that's was your latest paper was about.
Mm-hmm. Yeah.
What was the scope of that? Like, what sort of devices did you kind of test between the two types of 3D printers, essentially? And like, what were the results, essentially?
Yeah, so that goes back to the single piece sperm counting chamber. That type of device, the one of the reasons why devices like hemocytometers and macular chambers are so expensive is because they're they're precise devices. The reason you can calculate concentration of a sample on a device like that is because the volume underneath the cover slide is very precise. It's exact. And that has to do with the way it's manufactured. Special, like the weight of the cover slip on a hemocytometer is very precise. You can't just use any run-of-the-mill cover slip on a hemocytometer. They have special cover slips that are weighted properly so that the volume of the sample that you're counting is known, which allows you to do an accurate concentration. So when you're trying to 3D print something, there's a level of tolerance, or there's some tolerance that you have to achieve to guarantee or ensure that the volume underneath your cover slide is within some sort of known region, so that when you count the concentration, it's what you it's real. And so printing something like that on a consumer printer, it may be difficult, right? And so that's we have a CADWorks printer, right? And we bought it through a seed grant that Louisiana State University Agricultural Center provided to us. But the purpose of that paper that you were referencing was to see what the capabilities of consumer level and, in this case, industrial or commercial level printers are. Right? We didn't want to compare them because it would be an unfair comparison. If this is our scale of sizes that we're interested in, we wanted to know maybe consumer scale covers this much and industrial or commercial scale covers that much. And there's some overlap in the middle. And that was what was super important to us. Not all of the sperm that we work with is very, very tiny. And so the consumer scale printers are completely capable, what we found, completely capable of printing some devices for these larger sperm. I'll send you guys a picture of a variety of sperm, but ambystoma sperm are absolutely massive. We sort of call them like aircraft carriers. They are huge compared to human sperm, most fish sperm, even frog sperm. They're massive. And so printing on a consumer level printer is relatively easy when you're trying to achieve sizes for that scale. For zebrafish, which have sperm that are like a micron, maybe 2 microns big, like you're going to be hard pressed, or you may be hard pressed to use a consumer scale printer to create a device to count concentration for you, or to help you count concentration. And that is where having something like the industrial scale printer might, or commercial scale printer might actually be needed, because the size of those sperm are just so small that you have to create a gap that's small enough that you don't have a bunch of sperm stacked up on each other. So that was the purpose of the paper, was just to find where they overlap, and so that we can make informed decisions on when we create new devices in the future, knowing, oh, okay, we can create this on a consumer scale. It's going to be accessible to a lot of people around the world. Or we're creating this device and it's small, but it's not like soft photolithography small. And we can use a commercial scale printer and either print devices for people, or going back to that molding capability, print molds for people and let them pour a device themselves.
So now, after that, the paper that you had just written up, in terms of your, I think you probably have a significant amount of experience based on the wide range of printers that you've been using and the type of work you've been using. What do you sort of see in the future for yourself, your team, the new? Are there any new sort of features that you guys are hoping to develop for developing this repository?
I think having standardized devices are one place that we're really trying to push, and making sure that the communities that are using them, that they're serving the needs that the communities want. In terms of the printers themselves, I mean, faster printing times are always nice. Access to different resins. All of the printers that we have in our building are open material printers, which means that we can use any resin on the market. We just may need to dial in the settings a little bit, which again goes back to our philosophy of open hardware, right? We don't want to be stuck in one ecosystem. Just being able to control every aspect of the printer is great. And I think having more companies that allow you to tweak your printer to your liking is something that would be good at an even higher level scale than our center, right? That's like an industry level thing. But I understand that, companies need to make a buck. And so a lot of those printers that are closed material ecosystems, those companies put a lot of money into like dialing those printers in and making them work exactly as you expect. But there's also plenty of open material printers, both in fused filament fabrication and resin, that they just work out of the box because they're just workhorses, right?
And so, what is next for the germplasm repository? Like, what do you envision in terms of your work and I guess in any institution or team, there's always a changing mission statement. That when they sort of start attaining some of the goals or the milestones that they had set out, they start building brand new ones. And what would be the next ones look like?
I think our mission won't really change that much, because the number of communities and number of species is just so large that we could spend decades working on it. But where we may change is in how we enable other communities, and the speed that we enable other communities to create their own processes and repositories to address their own species, right? So that's like a distributed workflow, where rather than us being the only group cryopreserving species, helping to accelerate the number of groups that are cryopreserving different species, because we have expertise in a lot of different things, but there's a lot of other things that we just there's not enough time in the day or in our lives that we can do that. So we need more people. So getting the word out, doing more outreach. I think we sort of envision every middle, elementary, high school, every college student knows how to 3D print. You know, they're familiar with CAD, whether they're a accounting major, a psychology major, a nursing major, a biology major, an engineering major, because it's really a life skill at this point. Like everyone should know how to do that because you can do so much with it.
And so, because you're trying to build this wider community, where are where is kind of like your home base? Is on your website? So, say I was a aquatic researcher for a specific species, and I want to, you know, learn more about your cryo kits or anything like that. Would I find it just like, do I need to come across you based off your website, or are you on a forum or some other platform where, you know, AGGRC can be visible?
Yeah, so aggrc.com is probably the best place to go. We do have a community forum that we can give you an invite link to, that if people are interested in communicating with us and with other people in our community. Every time we publish a new open hardware device, it gets posted on there. Okay. And then we also have a variety of different social medias. I think we're on LinkedIn, Facebook, X, Blue Sky, Instagram, right? And so okay. I think there's messaging on those, so you could get in touch with someone here.
Wow. Right. Amazing.
So before we sign off, Jack, why don't you take a moment to give shout outs to all of those that have made your work and research possible?
Cool. Thanks, Hemdeep. Yeah, thank you so much to the Louisiana State University Agricultural Center. They provided some seed funding for some work that was very relevant to this conversation. A big shout out to the National Institutes of Health Office of Research Infrastructure Programs. They have helped us to help support us in our biomedical work, helping the national centers across the United States. And those include the National Aplysia Resource, the Xenopus National Resource Center, the Ambystoma Genetic Stock Center, the Zephophorus Genetic Stock Center, and the Zebrafish International Resource Center. Also, thank you to all of our collaborators at Louisiana State University, the Dr. Monroe's group, collaborators around the world, and other funding sources from USDA, National Science Foundation. So we were really appreciative of everything that those groups do for us and help us with. And we love helping people all around the world to protect the genetic resources of aquatic species.
Amazing. So what we'll do is that we'll add the links to all of those teams as well. I'm so happy that we had this chance to speak, and I think you guys are doing some amazing work, and I look forward to seeing some of the more interesting work as a technobiologist. I certainly am interested in seeing what you do next.
Thanks, Hemdeep. Thanks, Robin.
And I hope we get a chance to speak again soon.
That's good.
Take care.